level of individual variation, when you take multiple measures in the same organism, you're really going to be able to kind of understand their, and especially if you know what's happening in their environment at the same time, you're going to be able to see how they cope across time and how they're responding to that environmental change. So what this might actually look like in the field is I primarily do waterborne hormone sampling because I work with amphibians, and due to the ecology of amphibians, this is a really good technique to use with them because it's non-invasive and doesn't harm the animal. So I can capture my organism, quickly move them into a baseline sample, and all that is really they just hang out in a vial of spring water for an hour, and then following that hour, I can remove the animal and take the water back into my lab and process it and get a port value from that. Then I can move them into their agitation sample. So this is where I look at what their conditions are when they're stressed. And really that stressor is just shaking them 1 minute for every 3 minutes throughout that hour sampling period. While it's not necessarily the nicest thing, it doesn't harm the animal. And we have seen this generate a stress response in a lot of other amphibians in our lab. Following that, they go into a recovery sample to see if they're able to regulate themselves. Those conditions are the same as the baseline sample. So all in all, this assay takes 3 hours to look at their coping. So what I wanted to do was a multi-year individual level study of coping capacity, which we'll get at welfare, of wild lesser sirens, and I wanted to look across a lot of age classes. And ideally, we can kind of get a better understanding of the environmental pressures that these wild sirens are facing and be able to use that information to support their conservation. So this was a big project that was funded by the Wild Animal Initiative, and it was a collaborative project with a bunch of other labs. So we worked with Wichita State University. Tom Luring's lab came and did a talk a little bit ago, and he sent down 2 master's students into the field with me. And then SFA College, we worked with Montagna Lab, who sent out an undergrad into the field with us. And we all had different questions that we were answering with our project, but we helped each other with the field work. So I'm just talking about my component of that project. So we went out into the field and we were out there for 11 months. So I started in the summer of 2023, right after graduation. I was here for my master's, and I graduated, hopped in the car, drove down to Davy Crockett, and we started that summer, and then I came back, and then we went back out again in winter, spring, and summer of 2024. So it was 11 months in total. And for my question, we had 3 distinct populations that we knew sirens were at. That I used to answer my question. And because I was sampling these areas repeatedly across the 11 months, I tried to hit each population 6 times for 6 days. I wasn't always able to do that because there was stuff that was like out of my control. We'll talk about that in a little bit. But we did hit these populations a lot, and we saw them change throughout those 11 months. So there were pretty clear and distinct differences. This was like site 115. We had a lot of older, larger, mature, established sirens, and this was the site where I really got a lot of my repeated capture data. I was seeing the same guys again and again. There was 87. This population was mostly juveniles. There wasn't much else in this pond. I joked if you drained out the water, it would probably just be like a wriggling mass of sirens at the bottom, because the numbers were crazy, and you would just pull a minnow and it would be like wriggling with salamanders. Then my last site, 121, there weren't the same numbers that there were at the other 2 sites, but it was incredibly biodiverse. It was really fun to pull traps at this site. You never knew what was going to come up. We used a lot of different methods to get our salamanders, really however we could get our hands on them. We tried. So fikes, dip nets, minnows, and then we made these modified trash can traps by drilling holes at the bottom and placing funnels so they can swim in and they have a hard time swimming out, and they were effective. So we caught a lot of stuff while we were out there, and it was always super, super fun. I put this here for my herpetologists in the crowd because I thought you'd enjoy it. We got an amphuma. I'd never seen one of those before. Super, super cool. It was massive. There were cottonmouths in the traps, water snakes, bullfrogs, leopard frogs, cricket frogs. We got snapping turtles, musk turtles, red-eared sliders. It was super, super cool to pull traps every day and see what came out of it. Really amazing experience. But I had never had this kind of intensive field project before. I didn't have that much field experience going into this. So I definitely learned a lot. And my main advice, if you're doing a big field project, is much like the GC profile, you must be adaptive. Because stuff is going to happen that's out of your control. I was on public land, my fikes got stolen, they stole minnows too. You learn about the environment learns about you. So the snakes learned that when I set down my minnows that they could get a really easy meal, and it became snake buffet. So this is a snake that I pulled out of one of my traps, and I scanned him, and one of my pit tags from my sirens popped up. I had seen that guy a lot, so I was sad to miss him. Let's see, there were a lot of bugs. I wasn't able to use bug sprays because they breathe through their skin. That's annoying. There were road closures that blocked off the access to my sites because of flooding, prescribed fires that interrupted trapping, downed trees that interrupted trapping, flooded minnows in the spring was an issue. We hauled heavy equipment a long while. Morale got low at times. But in general, it was an amazing experience, and I'm leaving this project feeling a lot more confident and competent than I went into it going in, so I learned. The questions I wanted to know was, what does the GC profile look like in these siren populations? How is it varying across age and season? What does coping look like? Does it look healthy? And then how is coping getting affected by environmental change and things like rainy season and temperature fluctuations? So we went out into the field every day. We pulled traps, we recorded bycatch. If there were sirens, we scanned them for a pit tag, gave them an ID, and moved them into the water sample as quickly as possible, usually within 5 minutes. Then we took our 3 measures of port. Following that assay, everyone was measured for growth. We put a pit tag in you if you were big enough to get one, and then we took an ID for photo identification purposes and released the salamander towards where we caught them. In total, for my project specifically, we got 671 captures across the 3 populations. 70 of those were larval individuals, and that amounted to 2,013 waterborne hormone samples. I'll be running a subset of that, around 1,400 in the lab, and we got repeated captures on individuals up to 5 times. Not all the time, that was like the gold standard, but we did see individuals repeatedly often in our sampling. Okay, now I have to make the world's biggest bummer of a caveat. That many waterborne hormone samples takes a long time to process in the lab, and I am one person, so I'm still working on it. And I don't have all of the adult repeated capture data, but what I do have for you is the larval data. We weren't able to put a pit tag in the larva, and we couldn't reliably use photo identification because their markings change as they grow. So it's not repeated capture data, but I still do think we found stuff that's interesting and worth talking about here, so buckle up. Okay, so what does coping look like in our larval individuals? We saw that they went down from baseline and then went back up in recovery, so they went down in their stress samples, but they didn't significantly change across their sampling types. But what we did see was that their stress response, so if you remember, that's going from baseline to agitation, and their negative feedback, which is agitation to recovery, those were highly correlated with each other. And so what I want you to pay attention to, this is like a rate of change. It's not really giving you a direction of whether they went up or down. Same for negative feedback. But these points are clustering really nicely along my line. So if you had a high stress response, you did also have efficient recovery. If I had a lot of points that were like here, that would be indicative to me that maybe they're mounting stress responses or not recovering or vice versa. But they cluster along this line, so I took that as a good sign. That when they were stressed, they were regulating themselves, and to me that indicates that they are coping and they're doing well in the wild. As far as season, court was higher in the spring compared to summer. Lesser siren breeding season, they start to hatch in the spring. Court was also higher in smaller individuals than larger individuals. And then these are graphical interpretations of my models. I ran mixed effect models with population as a random factor. And how you interpret these are not means on the axis, they're beta values. So if it crosses the line, that means it's not significant. The farther away from the line, the larger influence it had on my model. And then if it's up, it's positive. If it's below, it's negative. So I know there's a lot of information. I'm basically like throwing tables at you, kind of. But I want you to pay attention to one key thing here. Oh, not that. Okay, cool. So for temperature, we had a positive relationship between temperature and court. When temperatures were higher, court was higher. But this was not significant in baseline samples, and it was significant in agitation and recovery samples. So I do think sirens may be vulnerable to environmental change, such as rising temperatures or drought, but they might be particularly sensitive to these under stress conditions. So what did we learn? Are they coping? So far, yes, it looks like the larval individuals are coping. They're decreasing in their stress sample, similar to the strategy a tadpole would employ, so that's interesting. As far as season, court is higher in the spring at the beginning of their breeding season, and temperature and court have a positive relationship. So I do think the meat of the information and the benefit we're going to get from the study is going to come from the adult repeated capture data over time. So I am sorry I didn't have that to present to you guys today, but ultimately I think we're going to be able to take these findings and really understand more about the subjective experience of wild sirens and be able to use that for conservation. And I think that's exciting. As far as future directions, I took some microbiome dermal samples while I was out there, so I want to look at that in conjunction with the court. I might do some immunology stuff in some later chapters, probably not with sirens, but I'm excited about it. And then there's actually another university that wants to do a behavioral project with sirens and like cue behavior stuff, and they asked me to help out on that, and I'm really excited to do some collaborations and stay in the siren world. But my immediate goal is to survive my proposal defense and finish college. This was a big project. I didn't just do this myself. There's so many people to thank. My lab, all of the labs, Tom's lab, Carmen's lab, the undergrads who came out into the field. Caroline was great that first summer, and then she left for another job. Shania helped in the field work. Anybody who funded me, Wild Animal Initiative, Texas Herb, Texas State, and then the tadpoles and the sirens. I couldn't do it without them. So does anybody have any questions?